Learning objective include classification of culture media, what is a pure culture and how a pure culture is obtained. Now, the media that we use for bacterial or microbial growth could be classified into three categories. They include chemical composition-wise, based on the chemical composition, based on physical characteristics, and based on the function of the culture media. Based on the chemical composition, the media could be defined and this is also synthetic. When we say defined, we know what ingredients we are putting in. This kind of medium is used when we know the nature of the bacteria, that this bacteria could grow in a defined medium. But most of the time, the nature of the media that we use is complex. And complex media, because the ingredients that are used in complex media, their composition, exact composition is not known. Like, for example, one of the ingredients in these complex media is meat digest, peptone, yeast extract. So in these chemicals, we don't know what kind of things are there, what kind of ingredients are there, but we know that they all support. They have growth factors in them, like vitamins, trace minerals, that are needed for those bacteria that are fussy or fastidious in nature, very hard to grow, very hard to please. Based on the physical characteristics, the medium could be either solid or could be liquid. Based on the function, the medium could be a general purpose medium. This is a medium that supports lots of microbial species, but not all of them. Second category is a selective medium. Selective medium or media are those that selectively support the growth of certain bacteria, and they would discourage the growth of other bacteria. Enrichment medium or media are used when we want to grow a particular organism or a species of organism. Then that organism is preferentially given a substrate or an ingredient that is used preferentially by that bacterium. Differential are media that basically we put different ingredients to differentiate between the two species. So I'll give you examples just in a short while. Differential medium, on the same medium, we can differentiate two different species of organisms. Like here, you could see this, the nature of these bacteria. This is basically a general purpose media. You can differentiate. There are two different kinds of colonies, but they are by nature. There is nothing this is a neutron agar plate. There is nothing that can selectively differentiate between these colonies. This colony that we see, yellow colony here, it is because the bacterium is able to produce a pigment. Exactly. This is the situation where when we use a differential medium, we could see the different colors of the colonies based on their ability to use ingredients differentially. This is a differential medium. It is also an enriched medium, neutron blood agar. Blood agar is prepared by putting blood, 10% blood, in the neutron agar. So basically, this is a basic medium, which we most of the time use, the neutron agar. When we add blood into it, it becomes blood agar. And blood has lots of growth factors and other undefined ingredients that support lots of bacteria or microorganisms. Definitely, it is used for fussy or fastidious organisms. Then it is also differential medium. It is enriched because of the blood, and it is also differential medium. There are bacteria like Streptococcus pyogenes. It has the ability to secrete toxins, which we call hemolysins. They cause hemolysis of RBCs. Some toxins cause complete hemolysis, as you can see here. This is called a beta hemolysis or hemolysin. And then there is an alpha, which is kind of partial. And then this colony here is non-hemolytic. That means that this organism is not producing any toxin that can lyse RBCs. So this is a differential as well as enriched medium. Another medium is a McConkey agar, very commonly used in microbiology. It is basically a neutron agar. Added into the neutron agar are bile, salt, and crystal violet. CV stands for crystal violet. Crystal violet inhibits the growth of gram-positive bacteria. and makes this a kind of selective bacteria for gram-negative bacteria. Bile also discourages the growth of all those bacteria that are not normally present in the gut. So this McConkey agar is usually used for those organisms that live in the GI tract, gastrointestinal tract of humans or animals. It's a selective medium because of these two ingredients, bile salt and crystal violet, and it's also a differential medium. For differential purpose, we put a carbohydrate, lactose, and then there is also a pH indicator, neutral red, that is also part of this medium McConkey agar. What happens is there are certain organisms that belong to the gut. They can use lactose, and when lactose is used, it is converted into an acidic product, and that acid changes the color of the pH. And those that are lactose fermenter, they would appear as red. E. coli is an example of those lactose fermenters. And then other bacteria that do not use lactose, they would appear normal without any color. So this is a differential as well as a selective medium. Now, what is a pure culture? A pure culture basically is a progeny of a single cell. So Robert Koch basically was the first scientist who developed this pure culturing technique. And with his technique, understanding of microbiology or this science grew significantly. So when we have a pure culture, we can study its characteristics. We can do diagnostic testing. So a pure culture is needed for lots of purposes. And Robert Koch made this technique useful. He used agar, which is basically a solidifying medium, and then he developed this streaking technique, which basically is essentially a dilution method. When you keep streaking, let's say we start from here, there are lots of bacteria sticking onto the platinum loop that we use, but as we go further along the line, the number of organisms would be reduced. So this is basically a dilution method. At the end, as you can see here, we started up here, then we sterilized the loop, and then we made a few lines over here, and then again we sterilized the loop because when we sterilize the loop, we flame the loop. The organisms that are sticking onto the loop, they die, and then we start afresh, touching these lines, because on these lines there are bacteria sitting on these lines. When we touch them, we drag those bacteria, and we basically dilute as we drag them. So this dragging and diluting technique ends up in single colonies at the end. Like here, you could see these single colonies. So single colony basically is representing a pure culture. Pure culture means is a progeny of a single cell. Now, as I mentioned, there's the streaking method is one method by which you can get a pure culture. There is another method, essentially is based on a dilution method or dilution technique, where what we do is we dilute the organism, like here. This is the sample, the original sample. You can make serial a 2-fold dilution, 10-fold dilution, or 5-fold dilution. So this is a 10-fold dilution here, and you can see that the number of organisms. As you can see, that this tube is much cloudy in nature, but as you keep diluting, the number of organisms reduce. So the idea is to get enough number of colonies that are progeny of single cells. So if we take these last 2 or 3 tubes and put them onto agar plate, you could see that as we dilute, the number becomes smaller, and there is more space between the cells. So these are likely the progeny of single cell. This colony, these colonies that you see, they are progeny of a single cell, and this is a purification technique. There are 2 methods by which we can use this dilution method to spread the bacteria onto the surface, like here. Or there is another method we will talk shortly, where you mix. Here you take the plate, which is already prepared, a solidified agar, nutrient agar, and you spread the organism, diluted organism, onto the surface of this. And there is another, what we call a pour plate, where we mix the diluted organism with the Newton agar when it is cool enough, but not solidified. And then we spread that pour onto a plate and then incubate. So that is, there are three methods so far we have discussed for getting purified colonies. One is streaking here. This one is streaking. The second is spreading using a solid agar. And then the third one is a pour plate where we use melted agar, but that melted agar is not hot enough to kill the organisms. So if we cool Newton agar medium to about 50 degrees that we can handle with our hands, bare hands, bacteria do not really die, and that method could also be used along with the dilution. So the first requirement basically is that we have to dilute the organism, and then either we can spread onto the plate or we can use a pour plate method. So in summary, media could be defined based on their chemical composition, based on their physical characteristics, or based on their function. There are general purpose media that support all different kinds and most of the bacteria. Then there are situations where we have to target a specific organism that require a specific ingredient for its growth, and that is called enriched media. Then we want to suppress the growth of contaminants sometimes, and we use selective media for that purpose. And we can, using the same medium, we can differentiate 2 or 3 different types of colonies based on their ability to use ingredients or not to use those ingredients there. And there are 3 different techniques. 3 techniques that we discussed for purifying cultures: streaking method, spreading method, and pour plate method.